So this will be a relatively high-level talk. I will be talking about AI technologies, I guess, in systematic reviews, and specifically natural language processing methods in this space. So I think, like a lot of times when I give a variety, like a version of this talk, I'll spend a fair amount of time sort of belaboring points about why evidence synthesis is important, and I suspect that for this audience, that's probably not, particularly needed. Nonetheless, I'll run with this this running example just to kind of start us off with some motivation, which is a very timely example of a particular clinical question one may have, which would be, sort of, all right, what treatments work well for COVID-19? And of course, if we want to answer this question in an evidence-based way, I guess the way that we might do this is we would consult, let's say, all of the highest quality evidence available to us. And, in general, for these kinds of clinical questions, the preference would be, if available, to consult evidence from randomized control trials. And I want to emphasize that because today's talk will really focus on, I'll sort of be focusing on RCTs specifically, in part because it provides a sort of grounding or semantics that means that a lot of the stuff we can do with natural language processing becomes a little bit more tractable. So I think that'll become more clear over the course of the talk. Right, so I'm going to be focusing on randomized control trials, and in particular syntheses of randomized control trials, right? So again, I imagine 90% of you are quite familiar, but the way that I think about what happens with an evidence synthesis is you're going to go out, you're going to find all of the published relevant evidence that's out there that pertains to your clinical question, and you're going to extract from these trial reports that you find the data that you'd like to synthesize. So for example, the reported efficacy of one treatment versus another, or what have you. And out of this process comes at least two things, or usually at least two things, right? So like a sort of narrative synopsis. So basically just a summary of what your impression of the literature is regarding this clinical question. And also, ideally, a statistical meta-analysis of some sort. Right, that basically puts together statistically the evidence pertaining to the different treatments, let's say for SARS-CoV-2 in this example. So in this particular case, if that were your question, you'd be in luck, because this is actually a rare case where the BMJ, for example, actually maintains what's called a living systematic review. In particular, it's a network meta-analysis that's actually providing an up-to-date, this is a slightly outdated slide, but it's continued to be updated as far as I know. And this is an up-to-date sort of synthesis of all of the evidence that's emerged for different treatments for COVID-19. So this is a very nice case, and they have this kind of nice up-to-date network meta-analysis where you can look at the relative efficacies of different interventions for treating COVID-19, right? So in an ideal world, for any clinical question that you would have, you would be able to kind of identify a living synthesis of trustworthy evidence in this way. And the reason that it's available in COVID-19 is because, it's extremely important and very timely topic, and it's obviously received a lot of attention. But unfortunately, again, this is the exception to the rule, right? And the reason that this is the exception to the rule is because producing and maintaining these things is just a tremendous amount of manual effort, right? So this is a paper that a picture that I took a long time ago now when I was first getting introduced to this space. So I was in the computer science department at Tufts, where there was also the evidence-based practice center. And this was like the set of abstracts that they were screening for just like one review. And this is sort of when I became aware of this whole problem. And this is also why I'm talking to you about this problem as a computer scientist and as somebody that does natural language processing. And the motivation, right, is that we have this somewhat insane system where the way that we disseminate the results of clinical trials remains primarily the unstructured natural language article, right? So somebody goes out, they do their trial, they disseminate the findings of that trial in a journal publication, right? Which is just not very efficient. And you have to have entire groups of people whose entire job it is to kind of manually wade through this literature and extract relevant evidence to synthesize into actionable information. right? And so again, like probably you're all maybe painfully aware of these facts. I don't know. But there's an estimate from BMJ 2017 that says it takes like 70 weeks to complete a single systematic review. So this is, by the time it gets published, it's probably already out of date. The rate of the sort of growth of the evidence shows no sign of slowing. So this is a figure where we're showing the estimated number of randomized control trial reports that are indexed in PubMed year over year. And we can see that it's, this was as of 2018, continuing to grow quite rapidly. And so for a couple of years now, my co-conspirator and myself, so this is Ian Marshall, he's at King's College London. We've been working on this project that we call Robot Reviewer. And the idea here is to have a suite of natural language processing and machine learning models that automatically can extract clinically salient information that would help those that are performing formal evidence synthesis. It's probably easiest to just see like a little demo. So here, this is the tool, the sort of prototype interface. There's a bunch of PDFs that are all describing clinical trials on the same topic, presumably. They get uploaded to the system. And in the background, we do a bunch of machine learning models. We apply them. And it does stuff like tries to generate a risk of bias table, like in the style of the Cochrane risk of bias tool, which I'll talk about in a bit. And also it'll try to extract snippets from the PDFs that describe key trial attributes. So things like the study populations, the interventions, and the outcomes. And you can kind of click through in this prototype interface to see from where in the PDF these snippets were extracted. So that's the idea behind Robot Reviewer. This is both like sort of everything that goes into this is open source and available. We also make the prototype available. So if you're interested in playing around with it, please do. You can also like run it locally if you're interested. And I should mention, just as like we were very happy to see this, like for the motivating example, like that BMJ study, they're actually using one model that's in Robot Reviewer, which is the randomized control trial identifier we have. And they're using this to help reduce the workload that's involved in keeping that COVID-19 study up to date. So that was cool to see. I guess more broadly, we have seen like some uptake of like Robot Reviewer and related technologies for systematic review production specifically. So the target audience here, I guess, are like evidence-based practice centers or you know, Cochrane groups that are actually producing these systematic reviews, I guess, for a living. So that's been really nice to see. And I guess what I'm going to talk about, though, a little bit more today is a slightly more general application of the same models that are in Robot Reviewer. So the way I think about Robot Reviewer is a set of machine learning models that can extract, I guess, various bits of information that are useful for making sense of biomedical evidence. And we've been thinking about taking that same set of models and maybe using it a little bit more broadly, so not just targeting systematic review producers. And this has led us to develop this thing that we call Trial Streamer. And the idea with Trial Streamer is that we monitor PubMed and some other sources of new evidence nightly. And what we'll do is we look for things that look like they're new reports of randomized control trials. And we use a machine learning model that we validated in previous work to do this. And we take the things that do seem like they're reports of new randomized control trials, and we run them through the models that I mentioned before. And these do things like extract the sample sizes, extract the PICO elements, so the descriptions of the populations, interventions, comparators, and outcomes, and also the risks of bias, as well as the main findings as best we can infer. And we dump all of that structured data that we automatically extract into a database. And this is sort of the Trial Streamer database. And so altogether, this constitutes like a living database that's updated daily of all randomized control trials, along with structured or semi-structured data that we automatically extract from these. And we make this available, both the raw database itself, which is a Postgres database for those of you that are interested in that sort of thing. You can download nightly dumps of that database and do whatever you like with it. And we also make a simple interface on top of this available at, which is this Trial Streamer interface. And This allows you to do faceted search over the entire kind of evidence base, or the entire set of randomized controlled trials anyway. Let's see. So if we look at an example, like here's what happens if you search for COVID-19 as a population. And this is what I mean by faceted search. You can search for particular populations of interest and particular, let's say, interventions of interest. So if you look for, COVID-19 as your population and let's say remdesivir as the intervention, it will retrieve for you all of the RCT articles that it knows about for these two things. And it surfaces, in this case, like the New England Journal article describing remdesivir. And I just want to highlight a couple of things that we're doing here. So first, the way that we're performing this kind of faceted search, where you can search for specifically individual PICO elements like populations or interventions or outcomes, is we automatically extract snippets from abstracts in this case. We have models that do it over full text as well, but here we're doing it over just abstracts that describe these elements. So we show them as well. Like when you search in Trial Streamer, it shows you the snippets that are that have been extracted for these PICO elements, and that's sort of how we index things. And the way that we do this is we train what are pretty much sort of standard sequence tagging models in natural language processing. And we train these on top of a data set that my student Bin Nai, shown here, collected a couple of years ago now. So if you're interested in this, the data set is available, and the paper describing it was published at ACL, which is a natural language processing conference, a couple of years ago now. This is what we've been using to train up our PICO models. And the data looks something like this. This is a simplified version, but basically for a chunk of text, you're going to have annotations that describe what snippets in that text pertain to the population, so like women with hypertension, the interventions, so like regular exercise or no regular exercise, and the outcomes. So in this case, it'd be hypertension, I guess. Okay, so I'm not going to talk about the specifics of that. It's a little bit older work, but these models are available, and so are the data sets if you're interested. We've also worked quite a bit on trying to assess the risk of bias. So in Trial Streamer, we actually provide a crude score that is, you can see as like sort of a proxy for overall reliability of the evidence. And this is estimated using risk of bias models that have been trained on Cochrane data. And we have a bunch of work on this in the past. I'm not going to get into the sort of technical aspects. If you are interested in that, please reach out or check out. This is an older paper now, and we do things slightly differently. But basically, we have a bunch of neural networks that first try to identify snippets in text that describe, let's say, how the randomization was done or how the blinding was done. And then using those snippets, we make a decision as to whether or not they seem to indicate low or high risk of bias. That's at a high level what we're doing. So like I said, we have prior work that I'd point you to if you're interested in the sort of technical details. I think for this audience, maybe what's more interesting is in the case of robot reviewer, where again, we're thinking about folks that are doing formal evidence synthesis, meta-analyses. We have built in the risk of bias assessment stuff into the prototype that I showed. And so this is maybe what the interface looks like, where on the right-hand side, you've got like a risk of bias form, the four sort of major, I guess, axes or dimensions that are defined in the Cochrane risk of bias assessment tool. So random sequence generation, allocation concealment, blinding of participants and personnel, and blinding of outcomes assessment. And we pre-populate these with predictions from the model that we've developed. And one of the things that we've done is we actually did our own randomized control trial to see if doing this actually helps people do risk of bias assessment. And so this was a lot of fun for me to actually like sort of not just look at like a metric that we would use in a retrospective like machine learning or natural language processing paper, but to actually see if the technology was helpful in practice. And basically the way that we did this was we recruited people that do evidence synthesis for a living, and in a randomized order, they were shown papers that they were going to perform risk of bias assessment on. they would always use the same interface, and either the form would be pre-populated with the predictions or it would not be. And they could accept or override the predictions as well. So the punchline here is that I guess the first order question is like, did it actually help them speed things up? And the answer is it does, not by a huge amount, but by about a quarter. And so, we think that's a pretty substantial gain, given that these things take a very long time to do. So this was nice to see. And we suspect that like if people got used to using the tool, that would actually deliver larger payoffs, because there is like a learning curve as well. But I guess I'm not showing it here, but the other thing that we looked at was sort of just the subjective usability of the tool. And we found that folks that are doing evidence synthesis really did find that this was, or they reported anyway, that they found the pre-population quite useful. Okay, so I've just sort of skimmed over. I wanted to kind of cover at a sort of high level some of the technologies that are in Robot Reviewer and I guess Trial Streamer. I'll talk a little bit in more detail about something we've been doing somewhat more recently, which is trying to actually infer the results that are reported in trials automatically. So one of the things that, for example, Trial Streamer will show you, and that's in the database, are what we sometimes call the punchlines, which are sort of snippets from the text that seem to convey the main findings, let's say, right? So in this case, it's a snippet that contains the reported efficacy or the comparative effectiveness, I guess, of this particular mRNA vaccine. And the way that we do this is we rely on another data set that we've collected. So a lot of the work over the years has been building up data sets with which we can train machine learning models, because there haven't historically been a lot of them. So this data set we call evidence inference. And the idea here is to have folks look at a full text article describing a randomized control trial. And then for a given intervention and comparator and outcome, what we ask folks to do is to say whether or not the intervention, for example, intervention A, significantly increased, decreased, or had no significant effect with respect to the given outcome as compared to intervention B. So that's the task. And I should say that this work was led by Eric Lehman, who's shown here, and he's now over at MIT, and Jada Young, who is a graduate student here at Northeastern. So that's the task. And in addition to just providing that classification, we also want to be able to surface a rationale or a snippet of text that supports that assessment. This is the punchline I mentioned, right? And one thing I should note here is that any given trial might look at multiple outcomes and also might make multiple comparisons. And so the answer that you want and also the rationale that you want will differ depending on which intervention that you're interested in, or depending on which outcome you're interested in. So that makes it somewhat challenging from the natural language processing perspective. So to collect this data, we actually, a lot of data collection work in natural language processing happens on something called Mechanical Turk, which is a crowdsourcing platform where you can hire up folks to do these micro tasks. So that's fine if you're doing something like movie reviews or something, but it's much more difficult for these biomedical texts, which are a little bit jargony. So in the past, for the Pico stuff, we actually did explore using Mechanical Turk with some success by collecting sort of redundant annotations and trying to be clever about how we aggregate those. But for this task, which is even in some ways harder, we actually hired folks that have medical training, and we hired them on this platform called Upwork. And you can actually find like people that have credentials and are actual medical doctors. And so that's what we did for this work. They're not super cheap, but they're also not super expensive. So it's kind of a trade-off, I guess. And so the idea was we first had somebody do what we call prompt generation. Do we have a question? Just background noise. Okay. So in data annotation, like the first step was to have people generate these prompts. So basically we give them a full text article that's describing a randomized control trial, and we're asking them to tell us what are some of the comparisons made in this trial. In other words, what are some of the intervention, comparator, and outcome triples, if you will, that for which results were reported, right? So that's kind of the first step. So for example, if there's a study that's looking at like Advil versus Tylenol, this is obviously a silly example, but if it's a study looking at Advil and Tylenol for headache pain, and maybe comparing both of these to placebo, then this might yield two ICO triplets, two intervention comparator and outcome triplets. One would be looking at the outcome is headache pain, the intervention is ibuprofen, and the comparator is placebo, and the other might be the other drug versus, so this would be like Advil versus placebo for headache pain as the outcome, right? And in this case, in both cases, the answer would be significant decrease. And I guess the punchline, the piece of support for this would be the same sentence that I'm showing here. So, right, so we have one person kind of generate these prompts, and then we have another person sort of separately give us an answer, given a prompt, given an intervention, a comparator, an outcome in a paper, tell us what the answer is, increase, decrease, no difference, and mark the support. And then we have a third person that sort of independently verifies these annotations. So that was the process. We've described the data collection process and initial models in a pair of papers in the last couple of years. And all in all, we ended up hiring 11 doctors for this annotation task. And this resulted in 12,600-ish annotated prompts spanning 3,346 unique articles. And I should say we're making use of the PubMed open access subset for this, which is nice because it means that everybody can have access to the data. Okay, so also the agreement here was quite high. So I think the annotation task is pretty well defined. So you can browse the data. Here's what it looks like. If you go to this kind of convoluted URL, you can sort of just kind of browse what the data looks like. And here's a real example where you have like ulcer healing as the outcome and compression therapy versus no treatment as your intervention and comparator. And I guess in this case, it's significantly increased. Okay, so now finally, I'll talk a little bit about modeling, not too much, but the way that we think about this from the natural language processing perspective is the model is going to be given at training time. We're going to be given a prompt, which is an intervention comparator and outcome, and a label, which is like significantly increased. That's a document level label. And then the input document and the highlighted span, so the piece of evidence that supported that. So that's going to be the training task. Let me pause and I see there's one question, which is how much did it actually cost? I should know that off the top of my head. I don't recall. It's in the paper. Off the top of my head, I don't recall, but I want to say like order 5,000, I believe, was like the total cost of the whole exercise. Maybe a little bit more because we did a second pass where we collected a bit more data, but not more than 10,000, I think. So something like that. Yeah. Okay, so this is like sort of from the modeling perspective, this is the information that we have, right? So we've got the full text documents, we've got the highlighted spans, the punchlines that are associated with each intervention, comparator, and outcome, and we have the associated classification, which is a three-way classification: significant increase, decrease, no difference. So in terms of modeling, we're doing something that's actually quite simple, but turns out to work very well, almost depressingly well, depending on your perspective on these things. So what we do is we have a two-step process. In the first step, we take an intervention comparator and outcome. We conditionally identify evidence that is relevant to that intervention comparator and outcome. And operationally, what we do is we just think in terms of sentences. So we first segment a full text article into the sentences that it comprises. And then we look at each sentence and we say, is this sentence reporting evidence for this intervention, comparator, and outcome? Yes or no? And then we take the piece of evidence that we find in this way, and we conditionally make a prediction as to the directionality of the finding: significant increase, decrease, or no difference. That's a three-way classification task conditioned on the evidence span and the intervention, comparator, and outcome. So that's what we're doing. In terms of like training, one thing this means is you have to train up your evidence identifier. And so to do this, we have our positive samples. We know what snippets were marked as being associated with a given intervention, comparator, and outcome. So to get negative examples, we just randomly sample other sentences. So these are very unlikely to report evidence for that intervention, comparator, and outcome because they weren't marked as such. And For those of you that are interested in this kind of thing, we're using what's now a very standard approach in natural language processing. So we're using this model called BERT, but in fact, we're using a variant called Biomed Roberta, which was introduced by some of the Allen AI folks. And this is basically like a big neural network that's been trained or pre-trained, if you will, on a bunch of scientific articles like PubMed, basically. And this is a variant of BERT that works a little bit better, basically. So if all of that is unfamiliar to you, don't like, basically it's a big old neural network that we're throwing at this problem, and it works like really well. So the second step is, once we've identified a piece of text that's relevant to a given intervention comparator and outcome, we then run that sentence concatenated with the intervention comparator and outcome through a different model also based on Biomed Roberta. And we then slap a couple of layers on top of this that make the classification as to whether or not this is evidence. One thing I should note here is, like, for the evidence identification step, you can do this sort of sharper negative sampling thing. But what do I mean by this? Recall that to train a discriminative model, we're going to need positive and negative samples. And as I said, you can just like randomly sample sentences and just assume they're not giving you evidence about the given intervention comparator and outcome. But you can also try to be clever. You can try to sample, for example, sentences that are positive for some other intervention comparator and outcome. So maybe a sentence that bears evidence for duration of headaches, but not for like mortality or something like that, right? And this is maybe a good strategy because those will look more like an evidence-bearing sentence in general, but at the same time will not be actually relevant to the specific intervention comparator and outcome that you care about. And so we found that doing these kinds of sharper negative sampling things seem to offer some modest improvements as opposed to just like randomly sampling sentences as negative examples. So in terms of the inference step, as I said, we just take another Biomed Roberta model, and now what we're doing is running that sentence through along with the sentence that we identify as evidence, the punchline, along with the intervention comparator and outcome, which are snippets of text themselves. We pass all of that through, and we just make a three-way classification on top of that, right? So yeah, it's the same model basically, and you just kind of encode the evidence, the intervention comparator and outcome, and you pass it through Biomed Roberta, and then a couple of layers that follow that, and then we just train the whole thing. And this actually works really well. So here I'm showing F1 on the y-axis. F1 is a standard metric in NLP. It's basically just it's a weighted average of precision and recall. And basically what this is saying is that on the left, the blue bar, we're getting nearly 0.8 F1, which is actually quite good for this task, in my opinion. This is macro averaged over the 3 classes. That's significant decrease, no difference, significant increase. And to the right, the other bar is showing the same model, but we cheat. That's why it's called Oracle. So this is where you assume that you're given the true evidence span. So basically, this is telling you, if you did a perfect job at identifying the actual evidence span, how well could you do? And you do get a bump in performance up to like 85 F1. So that model still isn't perfect, but there is room to improve both the identification and the classification, but these models actually do pretty well. So that's sort of like a pipeline approach, right? Like, it's not very satisfying because we just kind of say, okay, first I'll find the sentence and then I'll classify the sentence, basically. So something we've been working on recently is, you sort of don't want to do the pipeline thing, maybe. And also, like, there we're assuming that we're given the intervention comparator and outcome of interest. Of course, if you're just processing a new article, you don't know what the interventions, comparators, and outcomes are. So that also would be part of your pipeline, which would introduce its own errors. So one thing that we've been doing is sort of looking to combine like the PICO extraction task with the evidence inference task into a single model. And so that's the idea. So you want to go from like the raw text on the left to these like evidence frames that are basically, okay, this article looks at aspirin as an intervention, duration of headache as an outcome, and placebo as a comparator, and here's what it found. And you want to do that all sort of, all in the model, basically. And again, you want to basically be able to produce as many evidence frames as are relevant. So my student Ben, who also led the PICO work that I mentioned earlier, he's been working on this. And we had this paper at AMIA last year where we have an initial model for trying to do this. It's a somewhat audacious task. And the strategy that we used was basically to repurpose the evidence inference set, which has labels on for a given intervention, comparator, and outcome. And we took just the abstracts. So we're going to focus on just abstracts for now, and we'll ignore the rest of the full text in this case. So that gives us about 2,000 abstracts. And we had to kind of distantly label the interventions, comparators, and outcomes in the text in those abstracts, because the way that the evidence inference data set was collected, we have the interventions, comparators, and outcomes as free text. This is not an important detail, but basically we have a slightly noisy training data set where we've got basically, okay, here's a piece of text, here's an abstract of an RCT, here are all the interventions, comparators, and outcomes that were annotated, and here's the associated findings, basically. And so then what we did was we actually collected exhaustive, clean annotations on a test set to evaluate our performance of our model. This is a small set. It's actually very expensive to exhaustively annotate all intervention, comparator, and outcome triples and associated findings and evidence spans. So we only did this on a small set. So as a point of comparison, there's been a bunch of work in the NLP world on trying to do like this kind of joint modeling where you're looking to sort of identify entities of it within text. So in our case, entities would be the PICO elements, but you can think generally of like entities of interest, which are just text spans, and then jointly infer relationships between them. So one of the kind of popular models for doing this that's, I think, representative, let's say, of the state of the art is this model called DAGI++, which is, again, sort of a big transformer neural network that is trying to do these things jointly, and basically represented at the time of writing this more or less the state of the art in terms of how to do entity location. So that's finding the ICO elements, let's say, and inference about relationships between them. So deciding if the article is reporting that there is a significant difference or not, basically. So when you run DAGI++ over the training data set that I mentioned, collected for our purposes for this full end-to-end task of like, okay, here are the interventions, here's what's reported regarding them, you get pretty low looking F1 scores, right? So again, this is a measure that's a weighted mean between precision and recall. And you're seeing like 0.4, 0.45 maybe for the ICO identification task. And then in terms of doing the inference, like end to end, you see very low numbers like 0.2, right? So it's very hard. It's a very hard task. And so we were kind of thinking about how to improve this. And one of the things that we noticed just as a language thing was that you get this pattern a lot of times where, and this is a real example, where you'll notice that like in the punchline sentence or in the punchline snippet, which is underlined here, this pattern where somebody will talk about like the intervention maybe, but definitely the outcome. So they'll say something like erythromycin had little impact on reducing the outcome, low birth rate, I guess, or preterm delivery, which is a different outcome. And then basically they're only implicitly, they maybe don't explicitly invoke the comparator, right? Like they don't talk about placebo there, it's implicit. So we've noticed this pattern a lot where like outcomes seem to be emphasized in the punchlines. And so we thought about trying to actually exploit that observation. So what we do is we came up with this kind of approach that we refer to as ELI, which is short for extract, link, and then infer. And here's the idea. The idea is we're first going to go over the abstract, and we're going to use our existing PICO models and our existing punchline extractor to say, okay, here are the interventions, here are the comparators, here are the outcomes, and also here are the punchlines. And then what we do is we look at the most likely punchline, the most likely piece of evidence that seems to be reporting like the main finding. And we say, what outcomes are mentioned here? And that's kind of color coded in this kind of burnt orange. And then what we do is we look at all of the interventions and potential comparators in the entire abstract, and we score them with respect to how likely they are to be the intervention and comparator that is relating to the current outcome in the punchline sentence. So we train a model basically to do this, so we rank all of the potential intervention comparator outcomes and we argmax over those. And we say, well, I'm just going to assume that the punchline that I've extracted is reporting the relationship for this outcome between the top ranked interventions and comparator that I exhaustively enumerated and scored. And then we do the standard inference thing. So then given that we've identified the punchline, the intervention comparator and outcome, we can just do the standard inference step. That's part of the evidence inference task. So if you're interested in this, I'd point you to the paper, but that's the basic idea. And again, this was work that was led by Ben. And so this works a little bit better. It's still not very good. So this is still a very challenging task. And so there's still definitely work to do, but it does improve upon basically like the state of the art for doing this sort of thing. So sort of a mixed bag, I guess. Okay, so the last thing I want to kind of mention about this suite of tools is that one of the things I did over the summer and my rare opportunities to actually write code myself, which I unfortunately don't do very much of anymore, but I did port all of these models into something that's quite easy to use. So if any of you speak any Python and are interested in maybe using some of the models that I've been describing, we have made a suite of them like easy to install. That's the key thing. So you can just like pip install this thing RRNLP, and then you can use it. So then if you give it this is showing like an example call. If you give it a title and an abstract of a randomized control trial, like is shown in that little code snippet, then what it will do is it'll give you back like a dictionary that contains basically all of the stuff that we extract that we can extract from that trial report. So if you're interested in this sort of thing, then try it out, see if it's useful, and give us some feedback. Why would you do this? I guess like one of the interesting things that we hope and think people can do with both the trial streamer database, which we've already included all of these annotations, but also maybe the models themselves, is meta science. And in particular, you can take, for example, these 700, I think it's actually 760,000 randomized control trials now, and sort of do various analyses on top of them. Right. So one of the things that we've done recently is looking at what we think of as like disparities in the evidence or potential disparities. Right. So because we extract the populations, basically conditions for each trial that's in trial streamer using our models, we are able to sort of total up how much evidence is out there for different conditions. And we can actually go further than just looking at the number of studies, because we also extract sample sizes, so you can estimate the number of participants in total for each of these. So one of the things that we looked at was to say, well, how well, and if you look at, it's a simple and slightly naive analysis, but you might expect a priori the amount of evidence for a given condition would correlate with the burden, let's say, that it imposes globally. And so we looked at the disability adjusted life years as a proxy for this for different conditions. And we looked at how much evidence was out there for each of these conditions. And if you do just a simple regression where you, I guess, predict the estimated number of patients involved in all of the trials for a given condition as a function of the disability adjusted life years that it imposes, I guess, as a burden worldwide, you do see a positive correlation. So that's, I guess, reassuring in some ways. But you do also, of course, see outliers, right? And so you see things like enteric infections, for example, appear to be, on the basis of this very simple kind of naive analysis, may appear to be kind of understudied comparatively. Like we have less evidence for enteric conditions than you might expect given the burden that these impose. And similarly for respiratory infections. Now, there may be good reasons. You don't always need an RCT for things, and so I'm not necessarily asserting that, you ought to see no deviations or something like that. But I think maybe it highlights potential disparities or potential gaps in the evidence where certain things maybe appear to be overstudied versus other conditions appear to perhaps be understudied. So we have this BMJ Global Health paper if you're interested. I should say one other group at least has done what I think is kind of an interesting piece of work using the trial streamer database. So they were looking at methodological quality of a large set of randomized control trials using the trial streamer database, because that information is pretty readily available. So you have to accept that the risk of bias assessments are going to be imperfect, but still you can do this at scale because you have it for everything. And so we thought this was kind of neat. Okay, so the last part of this talk, I want to talk a little bit about a direction we've been going in lately, which is to actually generate narrative syntheses of randomized control trials directly. So not just extract stuff from them, but actually generate the synopsis. So here, I think maybe, yeah, so if you just, this isn't publicly released yet because we don't trust it for reasons that'll become clear in a moment, but we've actually incorporated this summarization approach into a very beta version of Trial Streamer, where the idea is we have a system that can spit out a summary, a free text summary for a set of input articles. So you give it like, 10 titles and abstracts of randomized control trials on a topic, and it gives you a summary that looks like this. And this is a real example. So in Trial Streamer, what you can do is just, you do your search and you take the top 10 results and you run it through this system and it will spit out the summary. And in the case of like COVID-19 and hydroxychloroquine, I think it actually does something reasonable, but it doesn't always do something reasonable. So how have we done this? Well, the task is pretty straightforward. What we're going to do is we're going to say we want to map from articles to summaries. So that in NLP parlance, we would think of this as just an instance of multi-document summarization. And it turns out we're in sort of a funny place in NLP, where in the past 2 to 3 years, we've all of a sudden gotten really good at generating text. So I think if you had asked me 5 years ago, in terms of being able to generate like fluent text, we weren't so good. But now we can really do it. So it's an interesting time. And what we're going to do more concretely is we're going to use the Cochrane data set again. And they have these author conclusion subsections, which look like this. And that's what we're going to treat as our target. So we're going to try to like generate those. Great. So we have about 10 abstracts on average for each of these reviews. And we've got order of 5,000, well, okay, more like 4,000 in total. And so only about 3,600 for training. And again, that's, so you have 10 articles, each instance is like 10 articles in an author's conclusion target, if you will. And so in terms of models, what we're using is something called BART, which is, again, big neural network. The idea is that you've got this bidirectional encoder. If you're familiar with BERT or similar, it's very much like that. That's like, you run your input text, in our case, a set of input articles through this encoder. And then you're going to hand off the resultant representations to a decoder, which is autoregressive. What that means is this left to right. And so it's appropriate for generation tasks. And so that will generate text like one token or one word at a time. Okay, that's the general idea. So this was work done by other people. We're just kind of taking BART as our starting point. Right, so in our case, we're going to pipe through our abstracts and we're going to output the author's conclusion section. One of the things that we did was we took BART and we continued to fit it. We continued to pre-train it on PubMed Central. So we attempt to map from article bodies, the body of full text articles, to the abstracts of those articles as best we can. That this, obviously you don't need any additional labels for, that's kind of free. And so this is why we were trying to do that. And we thought this might improve the performance of the model for our task. The other thing that we did was we looked at, well, look, we have all this extracted information for the individual trials that I've just been talking about for a while, things like the punchlines or the population descriptions and so forth. And so maybe we can integrate that into this neural summarizer, right? But it's kind of not obvious how to do that. So the way that we've come up with to integrate that information is very simple. What we do is we take the raw input, which might look something like this, where you've got like a title demarcated by T, followed by an abstract, which we mark by abs. And then the next article is you split up by a special token that says it's an S here. And basically that's saying, okay, here comes the next article, right? Because this is multiple articles. And then you've got the next title, and then you've got the next abstract. So what we've done is just extended that kind of demarcation strategy. So now we have special tokens, special words, if you will, that say, okay, here's an outcome, right, according to the model. So you take our RNLP and you say, hey, here's a snippet describing a study outcome, and then you mark that up basically in the text. And we do the same thing with the populations and the punchlines and so forth. And the intuition here is that because these are special words, the neural network will learn sort of to associate those special words with the fact that those are populations, interventions, or outcomes. And in a sense, what you're hoping is that by doing this, by explicitly demarcating these, you're freeing the model up from having to learn this implicitly. The only other thing I'll mention here is that we also have this limitation, which is these modern neural networks, they have a budget of words that you can fit. I wrote token here, that's like a word budget. So we have a bunch of, if you have 10 titles and abstracts, you probably have more than 1,024 words. And these models can only off the shelf, for memory reasons, basically, deal with 1,024 words at a time. So this is changing, by the way, like in the last year. So we're working on that now. But at the time, this was sort of a limitation. And so there's a question of like, well, given that you can't fit all of the inputs into your model, which of them should you pick? And so what we do is we prioritize including large and high quality studies. And we can operationalize this because we extract sample sizes and we have our risk of bias estimates. So if you give me 10 studies and I can't fit all of them in my neural network to spit out a summary, what I'll do is I'll drop small studies, or I'll drop studies that seem to be of low risk of, that seem to be of high risk of bias. Okay, so in terms of evaluation, the first thing that you look at, it's very hard to evaluate generation systems like this. If you use automated metrics like Rouge, you look at these different, I didn't get into the sort of nitty gritty of the different strategies, but the x-axis is different summarization strategies, including like, do we decorate, do we sort, like I just mentioned. And this Rouge thing is like an automated evaluation metric. And according to Rouge, there's no difference between them. But we don't really trust Rouge. In particular, Rouge doesn't say anything about how accurate these summaries are. It's really just a measure of like word overlap between a reference summary, so like the Cochrane author's summary, and what you generated. So we did some manual evaluation. So here's what the slightly low budget annotation interface that I put together looks like. We show a system summary, the summary that our model generated. And then we ask folks to sort of say if it's relevant and if it's fluent. And then we show them also the reference summary, so the summary that was written by the original review authors. And we ask them to assess, is the system summary factual with respect to this, basically? Like, is it reporting the same things? And it turns out that if with respect to relevance, they look pretty much indistinguishable. Also fluency, so these all seem to be like grammatical and they read well. They read as if a human wrote them. With respect to factuality, though, this is where which we don't know how to assess automatically. This is where you see the difference, right? And this is why I mentioned at the front that we don't trust these things. So in general, only about 40 to 50% were deemed like very to highly factual, meaning that the models tend to like hallucinate, to introduce things that are not true. So this is like a very big problem if you think about this particular application space, right? Like it's completely useless to generate narrative syntheses of evidence if you don't trust what these syntheses say. So, so this is like a nice challenge, I suppose, for us in natural language processing going forward, is to address this issue. And one of the things that we looked at doing was automating factuality scoring because it's not obvious how to do. And so for this, we have looked at using our punchlines. So you can take a punchline that's from your generated summary. We extract this with our model, and we also extract the punchline from the reference summary. And then you can make predictions with our inference model as to what is the directionality of this reported finding. And then you can compare. We're comparing using a symmetric KL divergence, but it doesn't, like basically it's just a measure of like how much these things agree. And you can say, okay, if they agree, then it's factual. And if they disagree, there's a problem. And this does correlate statistically significantly with human assessments of factuality, but the effect size is like the correlation is weak. So it's not a very strong correlation. So more work to be done, I guess. Okay, the last thing I'll mention is we're working on updating systematic reviews. So basically the idea is to allow folks to upload a systematic review when they first compile it, right? And for example, like a COVID vaccination review, and then we train a model, and it monitors trial streamer. And when so we use our model to recognize when new trials eligible for this review are published, and then we email them and we say, hey, you should probably look at these new studies. And we have a little interface for doing that. And then we offer like a bit of an interface for them to update the review. And we even try to integrate like the automated summarization stuff. And here I'm showing like the original review, and then the green and red is like a diff. It's basically showing like what the model, all the green is like the model thinks you should add this now because of the new evidence. So it's a little aggressive at the moment, but we're working on it. So we're hoping to get this out soon. We have a prototype, and we want to sort of allow people to use this. I'm not going to talk about simplifying reviews because I was too audacious in including new stuff. I will just mention that we are working on trying to also do simplification of medical texts, but I won't talk about it. So I'll just actually stop here for questions.
Great, thank you, Byron. That's an awful lot of work that you've got going on, and some really exciting new avenues. Nice to get some sneak peeks. Does anybody in the audience have any questions?
Hi, Dr. Wallace. I have a question.
Sure.
Yeah, thanks for the great presentation. Can you please help me? And I know nothing about machine learning AI. Can you please explain in simple words what do you mean by NLP?
Yeah, I mean, I'm probably not the right person to ask about this because like I'm somebody that came into NLP as like really a machine learning person that just happens to work with text. So for me personally, modern NLP is largely machine learning models applied to text, but this is not a general definition. There are also people that do what I might term proper computational linguistics, and these people are more interested in actually studying language using computational tools, or like formally modeling grammars and that kind of thing. And that's still a vibrant and important area of research. I happen to not really work in that particular area, but I guess NLP has morphed into a hybrid between sort of machine learning for text, but then also folks that are interested in like core real linguistics questions. Does that sort of make sense?
Partially.
Okay.
Thank you.
Sure. So I see there's a question that we frequently get, and it's a totally reasonable question. Have we done similar work on observational studies? We haven't, and we would like to. I'd sort of like to get it to the point where we can do everything with RCTs, just because, like, as I mentioned, just from a grounding standpoint, right? Like that kind of gives you this whole like Pico semantics, and just like the nature of randomized control trials lends itself nicely to kind of formalizing, I guess, in a way. So trying to branch out to other study design types was something we obviously would like to do, but just it's going to require a whole new data collection process, and it's good. We need new labels, we need new targets as well, right? Like we have to think about what that world looks like and what transfers and what doesn't. So basically no, but we'd like to.
Are there any other questions out in the audience? I had a question about the narrative synthesis generation and the low factuality scores. Is there a role for kind of having a human tell it, say this is why, this is where you're kind of going wrong, to improve the model?
Yeah, so definitely. So one thing that here I'll cheat and I'll look. So one thing that we've been doing is, in the case of simplification, and I'll cut to the end, we also observed these kinds of things where you have like hallucinations and stuff. So this is where we're going from a technical summary from Cochrane, because they have the technical summaries and then they have the plain language summaries. And it introduces these hallucinations or other things. And one thing that we've tried doing, to your point, is if you get a list of terms, in this case, we got them from, you could imagine getting this from humans directly, then you can actually try to tell the model, not to generate those terms. So basically, this is like this is in simplification. So basically, things that are jargony, confidence intervals, significance testing, and these kinds of things. And you can actually do something where you say this term. I'm not going to get into it, but basically, what this says is like penalize the model for generating those words. And so one thing that we've thought about is maybe if we collected annotations where people tell you that it's like, this is not factual, maybe you could kind of like use this similar technique to penalize the model for generating snippets that look like that. I don't know. But yes, but the short answer is I definitely think there's room for incorporating human judgments and in trying to use that to train the model. And I see there's another question about the low factuality because of the extraction of sentences and words. So no, I guess the reason I would say no is because we're doing abstractive summarization. So we're not actually doing extractive summarization. So in extractive summarization, you build a summary by just like taking snippets from the input and putting them together. But these models don't do that. They actually generate de novo, like new text. So I think one other kind of intriguing observation that we need to confirm, but is tantalizing, is like when you do pre-training on PubMed, like I said, it seemed, again, this needs to be confirmed, but it sort of seems like that might actually increase the amount of hallucinations. It decreases the amount of factuality, possibly because the model like learns to sound medically. It like learns to produce text that's like jargony and like reads correctly, but is just like not relevant to the particular input. So yeah, so I don't think it's not doing explicit extraction. So I guess I don't know, I think it's hard to say what's going on, I guess. And let's see, so yeah, so observational studies, yeah, I'm not sure. I need to seriously sit down and think about it. That'll be the next grant, I think. We'll visit observational studies next, I think. But yeah, so some of it might transfer, presumably would, but not all of it, I guess.
Great.
Oh, let me also give a shout out to my collaborators. Good, especially Ian, because Ian co-leads a lot of this stuff with me. How effective are transformer models in improving the state of the art? So I would say very. So the question is about like, how much does it matter, like these modern models? And the anecdote I would share is with the evidence inference task. If you look at our first paper at NACL 2019, it was pre-transformer, pre-like Bert and company. And we were getting F scores that were like in the 50s, 60s. And as soon as we moved to Cybert and then later Biomed Roberta, which are cousins of Bert, we are seeing these huge jumps in performance. So now we're up to like 80s or something. So really like in terms of macro average F1 anyway, for this particular task, it made a pretty big difference. In terms of pico extraction, we see less of a gain, like using for sequence tagging, like using a bi-LSTM CRF like works pretty well. I think you get a little bit of a gain shifting to BERT, but I've been impressed at how much these models have bought us. So yeah, so it'll be really interesting to see what the next couple of years bring, I guess.
Thank you. All right, well, I'm not seeing any more in the chat. I really want to thank you for your presentation. I think there's a lot of exciting things going on, and I'm sure there'll be people checking out your GitHub.
Great, yeah. So please shoot me an email if there's a there's a bunch of links, and shoot me an email if anyone has any questions or anything. I'd be happy to chat.
Wonderful. Thank you. I'm sure we'll be in touch.
Great. Yeah. Thanks again for the invite.
Yeah. Thanks. Bye.
Cheers.
Bye.